So let's start with next stage of Prophase-I, that is zygotene. Okay, so during this zygotene, pairing occurs between homologous chromosomes. So pairing occurs between maternal and paternal chromosomes which are homologous to each other. So pairing occurs between homologous chromosomes. Occurs between homologous chromosomes. Homologous chromosomes. So this process of pairing is called as synapsis or synthesis. This process of pairing is called as synapsis or it is also called as synthesis. Okay, let's see these chromosomes. So these are two homologous chromosomes which are, among which one is maternal chromosome and the other one is paternal chromosome. And these two chromosomes, maternal and paternal chromosomes, are homologous to each other. So after pairing, they are forming a structure called bivalent. Now this structure is called as bivalent because it consists of two chromosomes. And during this stage, zygotene stage also, each chromosome appears to be having only one chromatid. But originally, each chromosome consists of two chromatids, but it appears to be having only one chromatid. Okay, so now bivalent is formed due to the pairing between homologous chromosomes. Now this bivalent is also called as tetrad. Bivalent, or it is also called as tetrad. It is called as bivalent due to the presence of two chromosomes. And it is called as tetrad because of the presence of four chromatids. Because in each chromosome originally two chromatids are present. So it is also called as tetrad. Okay. Now the synapses can occur in three different ways. Let's see what are those ways. So the synapses can occur in three different ways. They are procentric synapses, then pro-terminal synapses, and third one is intermediate synapses. So in case of procentric synapses or pairing starts from centromere and it extends towards periphery. It starts from centromere and extends towards periphery. So this is called as procentric synapses. In case of pro-terminal synapses, this pairing or synapses starts from terminal, that is from the end of chromosome, and it extends towards, proceeds towards center of the chromosome, centromere of the chromosome. So that is called as pro-terminal synapses. Now coming to intermediate synapses. Intermediate synapses, in this case, pairing starts from midway and it extends towards periphery, that is towards the ends of the chromosomes and towards the centromere. So midway, what is midway here? So this is the end of the chromosome, this is the end, and this is centromere. The point which is present between the end of the chromosome and the centromere is called as middle point. So now this process of synapses or pairing starts from midpoint and it extends towards the end and even towards the centromere. So here also it starts from midway and extends towards the centromere and extends towards periphery. So this type of synapses is called as intermediate synapses. And the synapses occurs with the help of a complex called synaptonemal complex. With the help of a complex called synaptonemal complex. Synaptonemal complex. This synaptonemal complex is made of RNA and proteins. It also consists of very negligible amount of DNA. Very negligible amount of DNA. Hence it is also called as ribonucleoprotein complex. Ribonucleoprotein complex. Protein complex. Right. Now the synaptonemal complex exhibits tripartite structure. So it exhibits tripartite structure. Let's see what is this tripartite structure. Tripartite structure. So in this tripartite structure, it consists of three types of elements. One is central longitudinal element. Next, other type is lateral longitudinal elements. One central longitudinal element and two lateral longitudinal elements. And next type is transverse elements, which connects this central longitudinal element with lateral longitudinal elements. So this is central longitudinal element and these two are lateral longitudinal elements and these are transverse elements. Transverse elements which connect the lateral longitudinal elements and the central longitudinal element. This is the tripartite structure of synaptonemal complex, with the help of which both of these homologous chromosomes pair with one another. And the function of this synaptonemal complex is to stabilize these chromosomes till the end of crossing over. Until the process of crossing over gets completed, these chromosomes are stabilized with the help of this synaptonemal complex. So synaptonemal complex will be like this, in between these two chromosomes, like this. So it helps in stabilizing these two chromosomes, homologous pair of chromosomes, until the process of crossing over gets completed. When this process will be completed by the end of packeting stage. Next stage, process of crossing over will be completed. That we will study in the next stage. Okay. So here I would like to discuss one more point. So here we are calling it as bivalent or tetrad. So the number of bivalents or tetrads present in a cell is half the number of chromosomes present in that cell. Because one bivalent consists of two chromosomes. So to find the number of bivalents or tetrads present in a cell, we have to use this formula n by 2, where n is number of chromosomes present in that cell. Number of chromosomes present in that cell, not chromatids, chromosomes present in that cell. Let's say in this cell 4 chromosomes are there. So how many bivalents will be there? 2 bivalents. 2 bivalents or 2 tetrads. So what can we say? Number of bivalents is equals to number of tetrads present in that cell. Whereas the number of bivalents or the tetrads are half the number of chromosomes present in that cell. So these are the changes which occurs inside the nucleus of the cell during zygotene stage of Prophase-I of Meiosis I. Right. So what are the changes that occurs inside the cytoplasm of the cell during this zygotene stage? So already we studied in leptotene stage of Prophase-I, 2 asters are formed in cytoplasm and these 2 asters start moving away from one another, and in zygotene stage also they move further away from one another. So this is the change that occurs inside the cytoplasm of the cells during zygotene stage. So zygotene is marked for pairing between homologous chromosomes, formation of bivalents and tetrads. So this is all about zygotene. Next, let's proceed with the next stage of Prophase-I, that is packaging.